ELISA Enzyme-linked Immuno Sorbent Assay. It was first described by Enqual and Perlman in 1971. Solid-phase enzyme immunoassay to detect and quantify antigen or antibody. Principle: antigen bound to ELISA plate. Specific antibody conjugated with enzymes such as HRP reacts with the antigen. Addition of substrate result in colored reaction. Intensity of color can be detected by using spectrophotometer. This is the whole summary of the reaction.
Antigen is any foreign substance that stimulates an immune response. An antibody is a protein made in response to an antigen. Two types of antibodies used, monoclonal or polyclonal. An enzyme conjugate is an antibody joined with an enzyme. Most commonly used enzyme labels are horseradish peroxide and alkaline phosphatase. Substrate is a compound that undergoes change. Substrate binds to the active sites on the surface of enzyme and get converted by the change of color. Common ELISA substrates are PNPP, ABTS, OPD, and TMP. ELISA are classified according to the way in which the target antigen are captured and detected. Based on the capture, two methods, antigen capture ELISA and antibody capture ELISA. Based on the detection, direct ELISA and indirect ELISA. Apart from this method, there are two special methods of ELISA, competitive ELISA and sandwich ELISA.
In the first step, we take antigen from the sample and poured into multi-well microtiter plate and adsorbed at solid phase. After that, wells are rinsed off with PBST 3 times to remove all the unbound antigen, so only bound antigen will remain in the wells. An enzyme label, that is horseradish peroxidase labeled primary antibody, which is specific for target antigen, is added to wells and bind to antigen. After that, wells are again rinsed off with PBST to remove unbound enzyme conjugated antibody, so only bound antibody enzyme and antigen will remain in the wells. After rinsing, a substrate or chromogen like TMB, which is suitable for horseradish peroxidase that link to that antibody, is added. The conjugated primary antibody catalyzes the reaction with its respective substrate, resulting in visible colorimetric output. Color changes shows the sign of presence of antigen inside the sample. So if there is no changes, means there is no antigen found inside the sample. It can be measured by spectrophotometer or absorbance microplate reader. Direct ELISA is suitable for qualitative and quantitative detection in the sample of interest, antibody screening, and epitope mapping. Advantages of direct ELISA are: it is a simple and fast method. Just a single antibody required and workflow consists of only a few steps. It is less prone to error. Relatively small number of steps are there as compared to other ELISA method, and due to a single antibody, there is virtually no risk of antibody cross reaction. Disadvantages of direct ELISA are: antigen immobilization is not specific, resulting in potentially high background interference. Primary antibody must be labeled individually, which is time consuming and expensive method. It has a limited sensitivity, means there is no signal amplification step. It has a low flexibility, means a specific conjugated primary antibody is needed for each target protein. Immunoreactivity of the primary antibody may be adversely affected by labeling with the enzymes.
Another variant of ELISA is indirect ELISA. It is the most popular form of ELISA used to detect specific antibodies in serum sample. For doing indirect ELISA, we need two microtiter wells. In the first step, specific antigen to the target antibody are immobilized in the microtiter wells. antigen added to the wells are known antigens. The wells are then washed to remove unbounded antigens. In the second step, serum sample are added to the wells. First serum sample contains antibody of interest bind to the specific antigen coated in the well. While in the second well, without antibody or non-specific antibody will not bound to the antigen. Wells are again rinsed to remove unbounded antibodies. The antibody which is directly bind to the antigen is called primary antibody. In the third step, enzyme-linked antibody which is specific to the target antibody is added. First well contain primary antibody binds with enzyme-linked antibody. Since the second, there is no primary antibody and enzyme-linked secondary antibody remain unbounded. These unbounded antibodies in the second well are removed on rinsing. In the final step, substrate specific to the enzyme in the secondary antibody is added in both wells. First well with secondary antibody, enzyme act on the substrate to elect a chromogenic or fluorescent signal. This is positive test. While in second well, no enzyme-linked antibody, hence no color development. This gives a negative test.
Sandwich ELISA is used to detect the presence of specific antigens in the sample and also quantify them. Suppose if we want to detect the presence of A antigen in a serum sample using sandwich ELISA, for this we require antibody specific to antigen to be detected. So we require anti-A antibodies and enzyme-linked anti-A antibodies. In first step, a microtiter well is coated with anti-A antibodies. After this, sample containing A antigen is added to the well. A antigen from the sample will bind to the coated antibodies. Non-specific antigens will not bind to the antibodies. These unbound antibodies can be removed with the help of washing. Then enzyme-linked anti-A antibodies are added to the well. These antibodies will bind to antigens which are already bound to the coated antibody. So as you can see, the antigen is sandwiched between two antibodies. That's why the technique is called sandwich ELISA. And then chromogenic substance is added to the well. Enzyme bound to the antibodies will convert these substrate into the colored product. This development of color indicates the presence of A antigen in the sample.
Competitive ELISA is also known as inhibition or blocking ELISA, which is the complex ELISA technique of all. And its uses. It is used to determine the concentration of antigens or antibodies in the sample. Then coming to its principle, the sample antigen or antibodies competes with a reference for binding to limited amount of labeled antibody or antigens. I will explain it through the process of competitive ELISA. Then the result of this ELISA is inversely proportional to other ELISAs, by which if the concentration of antigen or antibodies in the sample is higher, means the color intensity will be very low. Next, coming to the process of competitive ELISA, here we are going to detect the concentration of antigens. For this, first we have to coat the ELISA plate with antibodies. After that, in the one well, we are adding the sample containing lower concentration of target antigen along with the enzyme conjugated antigens. And in the another well, we are adding the higher concentration of target antigen along with the enzyme conjugated antigens. After adding, there will be a competition between the target antigen and enzyme conjugated antigens. In the first case, there will be a higher binding of the enzyme conjugate antigens to the antibodies due to the sample containing lower concentration of antigens, but in the second case the sample containing higher concentration of antigens, so the target antigen will bind morely to the antibodies and enzyme conjugate antibodies will bind lessly. For the color reaction, we have to add the substrate. After adding the substrate get binding to the enzyme conjugate antigens and produces the color intensity. In the first case, we can see the higher color reactions because there is a low concentration of target antigen, but in the second case, there is a low color intensity because there is a higher concentration of target antigens binded to the antibodies. This is all about the competitive ELISA process. Next, coming to the result interpretation of the competitive ELISA. For this, results were interpreted with the standard curve. In the standard curve, if the absorbance value is higher, there will be a lower concentration of target protein. And next, coming to its advantages, the competitive ELISA are highly specific, suitable for small antigen samples, and the crude samples can be used for detection. This is all about competitive ELISA.
In this segment, we are going to see about the different detection strategies for ELISA. The final stage in all ELISA systems is a detection step. Detection can be done by a chromogenic assay or chemifluorescent assay or chemiluminescent assay. Now let's discuss all the assays one by one. Coming to the chromogenic assay is the most common detection strategy for ELISA. Enzyme conjugated antibodies are used in combination with chromogenic substrate solution, which results in a color change. The optical density of the reaction product is typically proportional to the amount of analyte being measured. This assay requires a spectrophotometer to detect the color change between enzyme label and the substrate. It sends a wavelength through each well to determine the optical density reading of the samples. The OD value is directly proportional to analyte concentration, as you can see in the illustration. Coming to chemiluminescent assay. Chemiluminescence is the light produced by a chemical reaction when the chemiluminescent substance is excited by oxidation and catalyzes, forming intermediates. When the excited intermediates return to their stable ground state, a photon is released which is detected by the luminescent signal instrument. For example, luminol reacts with hydrogen peroxide, which gives an excited intermediate aminophthalate and ultimately results in the stable end product along with light emission. The illustration shows the nature of reaction before and after adding hydrogen peroxide. Note the brilliant chemiluminescence after addition of hydrogen peroxide on the right bottom. Chemiluminescent assay requires a microplate reader that measures luminescence. The light given off by the reaction in wells is transmitted by a fiber optic cable to a photomultiplier that outputs relative light units. RLU is directly proportional to analyte concentration. The illustration gives a chronological order of events involved in the detection. Chemifluorescent assay. This assay is also enzyme based, but the generated product is fluorescent rather than colorimetric. Fluorescence occurs when certain molecules called fluorophores or fluorochromes absorb light. The light absorption raises the energy level to a brief excited state. As they decay from the excited state, they emit fluorescent light. A fluorometer can read the signal in relative fluorescence units, which is directly proportional to the amount of analyte being measured. Standard curve for quantification of ELISA. A standard curve is used for quantification of analyte. It is prepared by serial dilution of a known standard and plotting the mean absorbance in y-axis against the antigen concentration in x-axis. The concentration of target antigen can be quantified by plotting the values against the standard curve as shown in the illustration.
Advantages and disadvantages of ELISA. There are various types of ELISA present: direct ELISA, indirect ELISA, sandwich ELISA, and competitive ELISA. Coming to the advantages of direct ELISA, it is a simple protocol, time saving method, reagent saving, and no cross reactions from secondary antibodies is seen. Disadvantages of direct ELISA: it has high background, no signal amplification, and it has low flexibility. Advantages of indirect ELISA: signal amplification, and it has high flexibility so that some secondary antibodies can be used for various primary antibodies. Disadvantages of indirect ELISA: it is a complex protocol compared with direct ELISA, and cross reactions from secondary antibodies were observed. Advantages of sandwich ELISA: it is highly flexible, highly sensitivity, and highly specificity since different antibodies bind to the same antigen for detection. Disadvantages of sandwich ELISA: the antigen of interest must be large enough. It's sometimes difficult to find two different antibodies that recognize different epitopes on the antigen of interest. Advantages of competitive ELISA: high flexibility, high sensitivity, best for detection of small antigens even when they are present in very low concentrations. Disadvantages of competitive ELISA: relatively complex protocol than others, needs the usage of inhibitor antigen.
Some of the applications of ELISA are as follows. The presence of antibody and antigen in a sample can be determined. To determine the concentration of serum antibody in a virus test. During disease outbreaks, it is used to evaluate the spread of disease. It is used in the detection of antibodies in a blood sample for past exposure to disease. It is used in the determination of serum antibody concentrations in a virus test. Screening of donated blood for evidence of viral contamination. It is also used to measure hormone levels, as in the case of pregnancy tests. It is used to detect infections like HIV, syphilis, and other sexually transmitted diseases. It is also used in the food industry to detect any food allergens. In aquaculture, ELISA test has been used to detect Aeromonas salmonicida in fish tissues, clinical cases of enteric redmouth and furunculosis in fish farms, and Vibrio parahemolyticus in pinnet shrimps. ELISA has also been developed for rapid detection of viral hemorrhagic septicemia virus and striped jack nervous necrosis virus. Indirect ELISA can be used to detect gill rot disease, infectious pancreatic necrosis virus, and turbid hemorrhagic virus. Sandwich ELISA can be used to detect grass carp hemorrhage virus and infectious hematopoietic necrosis virus. ELISA is now a very useful and established laboratory technique in clinical medicine as well as basic translational and clinical research. Many variations of ELISA exist, including sandwich ELISA, the indirect assay, and the competitive assay. The choice of assays depend on the particular purpose of the ELISA, as well as other factors such as the size of the analyte, presence of non-competing epitopes, and the availability of the purified antigen. In fact, every single ELISA has to be designed individually based on the exact goals of the experiment and the characteristics of the antigens and/or antibodies that one is working with. Besides its other uses, ELISA is widely used in disease diagnostics and drug screening.